I am going to present the first long sequencing platform present in Argentina. We have established the first long-read sequencing platform applied to One Health at the University of Buenos Aires, operating within the School of Science, which is a free and public institution. The accessibility technology developed by Oxford Nanopore allows Argentina to have next-generation sequencing long-read equipment for the first time. We perform several applications addressing several biological questions, such as metagenomics of water, transcriptomics of human rare disease, and genomics of animal pathogens. Since 2020, we addressed several questions, and today I will focus on genome skimming of parasitic nematodes. The nematode we study is Dioctophyme renale, known as a giant kidney worm and considered the largest parasitic nematode of terrestrial vertebrates described to date. It's a health problem in dogs and wildlife, and also for humans, population in riparian areas. It's hard to sample because it's obtained from surgeries or roadkill wildlife, and there is little molecular information. We propose a genome skimming strategy, which is a low-cost and robust strategy to assemble mitogenomes from difficult-to-obtain specimens. It's useful for studying mixed samples and taxonomic groups without reference genomes, such is the case of Dioctophyme renale. We established this workflow, sampling, and library construction with ligation kits. We sequence all the genome, nuclear and mitochondrial genome, and assembly the complete mitochondrial mitogenome very quickly. We annotate it with custom databases and designed new mitochondrial molecular markers. Then we sample parasites from Argentina and Brazil, from domestic and wild species, and perform amplicon sequence using these new mitochondrial molecular markers, and determine that wild and domestic hosts share parasite populations, and also that parasite samples from South America are genetically closer to each other than those from other regions. Also, we describe unique features of nematode mitogenomes. We describe a new flanking non-coding region flanking each gene of the mitochondria, and the presence of non-coding region flanking each gene produces some context with alternative gene orders. These parasites may not have a unique mitogenome structure, so comparisons of the mitochondrial organization across species reveal that variation in gene order and in non-coding region copings is a common feature in this nematode group. For conclusion, I want to say that real-time long-read sequencing allows us to assemble and annotate a new mitochondrial genome very quickly. Without the reference genome, no mitochondrial DNA separation required, no amplification needed, and all mitochondrial genes were annotated, and we developed new molecular markers very quickly and identify unique nematode mitochondrial features. Very thanks to all my group, especially to Agustin and Natalia, who performed the sequencing and the assembly, and to Professor Gisela Francini, who is the co-leader of the genomic project, and especially thanks to Natalia Cortez from Oxford Nanopore, and all the staff of Oxford Nanopore here in Boston. Thank you.